Aha. The mic was muted. That'll do it. Anyway, let's see. I'm waiting for Otangelo to show up. I'm not sure if we had a miscommunication or not. I think he's still like live streaming on his channel. Hang on a second. Let me check that out. And this is going to be pretty disorganized. Until I figure my stuff out here. Hi, Leo. I think we'll have a little entertainment until then. Let me see where I was at.
Now, you have to find out first what makes more sense. How can we explain our existence and origins better, in a better manner? What is the evidence? How is the evidence better adequately explained?
Well, I have a completely different view. Like I said, I mean, you look at the history of science and just all of what we have done to study the universe. Take a look at the universe. I mean, basically,
Actually, I'm going to go out here.
Machine designer is what has given me a background, easy to understand what happens in molecular biology.
So you got to go.
Okay, so I guess where I was trying to go with this is, now you believe your God is obviously outside of the natural world, correct?
Yes, God transcends the natural world. He's in another dimension.
So if you're searching...
hello, Otangelo.
Hi, Mike.
Hi. Did you stop your stream on your channel?
Yes.
Okay. Yeah, a little bit confusing. I'm not sure how much bandwidth I have right now, but anyway, we are live already, so I'm...
Okay, let me restream it to my channel then.
What's that?
Let me restream your channel to my channel.
How can you do that?
I'm trying. Let me check here.
Something you can do on StreamYard?
I'm trying. Let me check here if I can do it. Yeah, it's working.
Really?
Yeah.
I had no idea you could do that. So that's something in StreamYard that you can do?
Yes, it's the same thing as adding a YouTube channel to be screened on StreamYard. So I'm just taking the other window where we are in here at StreamYard and rechannel it at my StreamYard.
Okay. Okay, so I reviewed a little bit of your video yesterday where you were talking to Lori.
Okay.
And I was looking at your talk on methodological naturalism, and I think the first thing I found out that I had a problem with was this one right here. Let me play it. See if you can hear it okay.
That is something which I see very often. When I go to a chat room of atheists and they discuss why they became atheists, in most cases, I would say maybe 90%, they will go down that line of reasoning. I was a Christian and I did read the Bible, and then I started to see discrepancies and difficulties and contradictions, and things that sound absurd to me. And then they started a process of deconversion where they came to a point where they simply said, well, I can't believe this stuff anymore. And then they do not, as an alternative view, elaborate something that gives them a justification for materialism, but they just stop there and they become agnostic atheists.
Okay, I don't think that's the case, but that certainly wasn't the case with me. For one thing, I was never a Christian, so I always derived my epistemology from what I learned as a child and what I've learned since then as an adult. And I don't find it to be baseless at all. I find it to be very powerful and matches reality very closely. So I don't know what your criticism is here. Most Christians that I've talked to that have become atheists, it happened to them in college. I guess I've talked to a lot of scientists that have become atheists, and they became atheists because they started studying. They usually come from a young Earth environment, or they've been brought up at young Earth, and because of young Earth, when they started learning that young Earth was absolutely impossible in college, then they immediately abandoned everything and started to call into question everything that had been taught to them. So I think young Earth creationism is probably the single biggest contributor to atheists today. Are you a young Earth creationist?
Yeah.
Otangelo?
Yes, I am.
Okay, so you're a young Earth creationist. I think
yes.
that is probably the.
Yes,
Yeah,
yes.
I can. Yeah. Do we have a lag?
I don't know.
Okay. I'm not entirely sure, but you don't seem to be responding quite as quick as I would expect. Anyway, I'm not sure if we have a lag or not, but. So if we do, we're going to have to be careful not to walk on each other. But anyway, let's see. The foundation for naturalism, I insist, is what we know every day as human beings, and that basically we are all naturalists. And one of the things that I did is I was talking to Tony on Smokey's channel, and I wanted him to consider what happens when you plant a seed in the ground. And when that seed turns into an oak tree, you do agree that there's nothing supernatural involved there, right?
Yes, but the seed has already all the information to become a tree.
Right, but we're not talking about what happened before the seed came in. We have the seed now, and the seed has DNA in it, and it has a bunch of other molecular information in it, and it has a lot of cellular information. It has a proteome built into it. So I mean, all of that there, if we start right now and watch it turn into an oak tree, there's no supernatural intervention there at all. That's all naturalism, right? All natural processes. You would agree with that?
Sure. Yeah, of course.
Okay. So that is the basis of naturalism. What is happening currently right now that we can observe, like you talk about the difference between historical and what was your other name for the other?
Operational science and historical science, which make two different questions.
Okay. So for operational science, it is all naturalism, and that's how we do it. And we do it with methodological naturalism. So that was.
Yes. And I think that's fine. That's fine. That's okay.
Okay. So that is the basis for naturalism.
I don't think scientists should go and investigate the protein and then expect that something supernatural or a divine action happens during the enzymatic performance. That is, of course, not what one should expect and not what happens. But I make a distinction in regards of historical sciences because they ask a different question. There, the question is how did everything come to exist? And the problem which I see there is that intelligent design is a possible explanation, but it is not permitted a priori to be an explanation. And this is, in my understanding, a problem because whatever you investigate, you must give a natural explanation and excluding a priori intelligent design. And this is, in my understanding, a problem because intelligent design is a possible explanation. Then why not investigate it, or why not permit the evidence to lead to intelligent design if that is the case? Just because,
yeah, if we found it.
Just because the foundation since Tom Huxley and the X Club in the 1900s, they pushed methodological naturalism into science, and then were successful. And since then, science doesn't permit intelligent design anymore or creation as a possible explanation. This is arbitrary, and this is what I don't agree with.
Okay, I'm not sure if I agree with you that that's what we've done. I mean, there's always the possibility that the entire universe was created last Thursday, and we wouldn't. I mean, we have to consider that as a possibility, right? It could have been created last Thursday. How would we know?
Well, the only point that I make, Mike, is that why is science excluding a priori one of the possible explanations, which is intelligent design? And why not? We could even say, well, maybe it was created yesterday, and then, I mean, this is a silly proposition. A silly proposition, of course, and we can easily exclude it. But why a priori exclude one of the two, in my understanding, big possible explanations? One is intelligent design, and the second one is natural materialistic explanations.
I'm asking Leo in chat if he wants to have. You remember you and Leo were supposed to talk?
Yes.
Maybe we could do that now.
Yeah, no problem with me.
Okay.
If he wants to come in.
Yeah, Leo, if you want to have that talk now, but I'd like to spend a few minutes on this because I'm puzzling this out basically. Now you're suggesting that we look for.
Just a moment. I have to attend the call. Just a moment.
That's
please.
Way too happy.
Yeah.
Okay, so Leo disconnected for a minute. So okay, I'll send you a link, Leo. And we don't want to gang up on him, so we're just going to do this one at a time. I'm going to take a back seat. I won't take a nap or a shower like praise that I am, but maybe I should. Okay, but give me a minute. Are you back, Otangelo?
Yes.
Okay,
yes,
you're
I'm
back. Okay, you left. Anyway, let's see. So okay, we have like if we're considering history, like if I'm considering, like I have a flower, and I am considering how that flower came to be, then I am going to consider basically methodological naturalism. I'm going to look for natural means for that flower to have grown from that bud, right? So if I'm looking back one day, I'm not going to be looking for intelligent design yesterday, right?
Well, origins englobes all physical existence. That includes the origin of the universe, the Big Bang, fine-tuning, origin of life, origin of the chemical elements, origin of biodiversity, origin of conscience, of the mind, of morality. Whatever we know exists englobes that question: how did everything that exists came to be. And that's not only in regards of biology, it's in regards of all reality. And
Okay,
that's my point. That's my point. Intelligent design is a possible explanation. And
okay, let me pin you down on this right here. Now, I was talking about yesterday and the day before, and you went all the way back to the beginning of the universe, or there is no such thing as the beginning of the universe, by the way. Don't think that way. You went all the way back to the big expansion, and then you went and then you mentioned a few stops in between. So that is one of the problems I have with any dialogue with can I call you guys? You guys in quotes. That is a problem I have, that you won't sit still. You won't sit still. So far we're talking about a flower blooming, and we're talking about using methodological naturalism. And now, do you think that's justified? If I were to figure out the science behind how a flower is born, how it turns from a bud into a flower, do you think I have to consider intelligent design right there? Do I need to know that? Do I need to know?
If your question is, how is the developing mechanism working? How does the plant develop evo devo? Then you will try to find out what the mechanisms in biochemistry are. But that doesn't give you an explanation of the origin of the mechanism itself. These are two separate questions.
Okay, what if I don't care about that? I just care about how it went from bud to flower. That's the only part I care about.
Okay, you are entitled to do it, but if the question is of ultimate origins, of ultimate reality, and of the origin of development itself, and you just restrict yourself to natural explanations, you are excluding one of the possible explanations, which is intelligent design, a priori.
Okay, we've moved off the mark. Now I'm asking you again, do I have to consider that supernatural or intelligent design if I only care about, yesterday I had a bud, today I have a flower, okay, I have some more buds, I want to figure out how buds turn into flowers. Do I care about intelligent design, or am I going to use strict methodological naturalism?
That is a question in regards of how things work. This is operational science, and in that case, you just use methodological naturalism.
Right.
And I am fine with that.
So you agree with that? Okay.
Yes.
So what I'm going to tell you is that if I go one day before and I look at how the bud formed off of basically a stem where there was no bud before, I'm also going to use methodological naturalism. And now I'm going to go one day before to when the stem was not there and we just had a seed. Let's go 5 days before. And I just had a seed, so I'm still going to use methodological naturalism. Okay. Now you understand evolution. Now you understand evolution. And you understand what we're doing. Think about it for a minute. But I think I'm going to let Leo in, but I want you to think about what I just said.
Yeah. I am thinking about it, and I think that you moved from one question, which is how does development work, to another question, which is how did everything come to be in the past, which is a different question. Origins is not. Origins is a different question. You can move back and back and back, and then you come to the origin of the universe, and then you ask, but what caused the origin of the universe into existence? And then you have three possible explanations. One is the universe was not created; it has always existed. The second explanation would be it didn't exist; it came into existence, and it just popped up out of nothing. And the third explanation would be that something caused it into being.
Okay, now I want you to play this part back, because I'm going to tell you that what you've done I was talking to you. I explained to you what evolution is. I explained to you what our research is all about. And you took the goalpost, and you're the one that ran all the way to the beginning of the universe. You ran across the universe. You ran across time. You went 14.2 billion years back, or 13.2. And that is your concern, okay? This origins thing, this historical, you don't stay in one place. And that is the problem with the difference between my worldview and yours, okay? Oh, Dmiter is here too. You have two of them now. You want to talk to two of them at the same time. That's going to be interesting. But anyway, I just want you to think about what I just told you, that I was not talking about the origin of the universe. I was talking about how a seed forms. That is methodological naturalism. And if you extend that, I can go all the way back to the common ancestor of that plant. And I'm still in the methodological naturalism field. And I have not needed intelligent design at any point in this discussion. Okay, there's only when you grab the goalpost and you run across the field. And that is the problem I have.
And let me make this nuance, Mike. As I said before, asking what the mechanisms are which transform a seed into a plant or into a tree, which is evo devo, this is a question of operational science. How does it work? What do we see? What are the mechanisms that operate in the development of the plant? But how did these mechanisms come to be? And I can right away advance what my explanation is. It is not evolution, but it is pre-programmed instructional complex information and various signaling pathways in genetic and epigenetic mechanisms. So the question is, how did that come to be? And of course, then there come other questions into play. If it actually evolutionary fashion, stepwise fashion of development is even possible, and that's where I put the big question mark, because then come other questions. How was the information in these signaling pathways, in these epigenetic codes stored there? How did it go? How did it come there? And how did the information to have the first living organism come there to start self-replication?
Okay,
and,
that'll be that will be.
If you say just evolution, then there is a problem in my understanding.
That will be the second part of what I talked to you about. But I want you to consider what I just said and think very carefully about what I just said about methodological naturalism. And I'm done now. And I'm going to introduce you to Dmiter and Leophilius. Have you guys met before, either of you?
Obviously I know Otangelo, but I don't know Dmiter.
And you have to be careful. There's a bad delay, I think, with Otangelo, because I think he's streaming to another platform. But I'm going to mute my microphone.
I will mute it too. So I will just be at your platform, and then I'll stream it later on. Is that okay?
Mike,
I think it's better.
Mike, let's just check the microphone to see how we can listen each other, because I have a little bit difficulty to listen well, but I will try my best. Let me see with the microphone here.
I can hear you real well.
Mm-hmm.
Okay, now you're muted, Dmiter.
Okay, just let now.
Can you hear me now?
I can hear you now, but now you're weaker.
Oh, shit.
I'm weaker. Okay, just 1 second.
Where did Otangelo go?
I don't know. He changed something on his computer and then he dropped out.
Oh, he'll be back. Probably.
See, this is why when I get my channel going, I will not be using StreamYard. I will be using either Zoom or Google Meet and be going through Streamlabs, because both those platforms are way smoother than StreamYard.
Please teach me how to do that, because StreamYard is starting to suck for me too.
See, and I'm just going to get a hold of Nakasuchi, the Rage, because he does that, and then Steve McRae, because he uses Google Meet.
Okay, hold on. Dmiter, you want to test?
Can you hear me now? Can you hear me now?
Really well. Really
well. Okay, that's good,
good
because
sound. Very good sound.
it's much better, because right now it's better with all the speakers here, so it's easier for me to understand and to hear. Okay, and Otangelo is here.
Okay, be careful with Otangelo because I think there's a bit of a delay with him, so try not to walk on each other.
Well, let's test if there's a delay. Say something when I'm done talking, Otangelo.
Yeah. Okay.
Okay.
Okay,
Yep. Nope,
that's good.
He's good. He's good.
Yeah, I removed my channel, so I'm
okay. I'm gonna bow out
in your chat room now.
Okay. So Mike, you don't need to mute yourself. I don't know. I mean.
Oh, I really want to mute myself.
So
I'm gonna go make a cup. I'm gonna go make a cup of coffee. I think we don't want to gang up 3-on-1 on Otangelo, and I don't know if you've met before, but Otangelo is one of the nicest creationists out there, other than Sal Cordova. They're both they both deserve respect. Okay.
Oh,
so,
I realize that.
Okay,
I realize that this is one of the reasons that I actually wanted to have a chat with him.
Good.
Because I see that he, at least it's reasonable.
Yep.
Let me say, in my experience with the creationist disputes, there was several different interesting disputes with the creationists, and there was few of them was very nice, but most of them was not very fruitful, and they didn't come to any conclusion. So my hope is that we'll have a constructive scientific talk, not just not, I mean, you know what I mean, so not constructive. So Otangelo, can I, so actually, how is your name? So how you want me to call you?
Otangelo.
Otangelo is okay?
Yeah.
Okay, so Otangelo, I just trying to figure it out here before we to start chatting each other. So from what kind of biological background you're coming?
I'm a layman. I have no formal education. I learn everything that I know through the internet, through science books and science papers.
Yeah, I mean, that's good enough. See, in my own career as a molecular biologist, I've seen many cases, and even very frequently, amateur scientists actually know much more and much better than the professional scientists. I've seen that. So to me, this is not criteria of knowing, just professional versus not professional. And to me, the most important is what someone is, how he is thinking, what his point of view, and it's about the level to which he is going deep into the material, into the science. So I can share with you a few things that I experienced before with creationists, for example. Around probably back in time, maybe about 6 years ago, I was in a very intensive chat with a girl. She called herself I think was Gneiss. She was very strange type of creationist. So according to her view during that time, she was completely acknowledged the existence and agree with the existence of evolution. And to me, it was very difficult for a very long time to figure it out what actually her problem is with evolution. But she was a creationist. So after long chatting, finally I got it. And I figured it out that, yes, she acknowledged the evolution and all of the science behind the evolution. But according to her, at some moments during the history, she was also old world creationist. So, and according to her, at some point during the evolution, intelligent designer intervenes and puts some extra push
Yeah,
to specific
I know how
direction. So that was Gneiss. And she was very reasonable in debating. Also, and I told her, it's a very legitimate hypothesis. You can say that. You can provide any kind of hypothesis as long as you can try and figure it out how to test this hypothesis, and as long as it does not have gaps in the hypothesis. So, and I'll explain you a little bit later what does it mean that. I mean, you can guess, of course, but so, and I told her, well, if you have that hypothesis that intelligent designer intervenes, for example, from time to time, to prove that, you're supposed to find a DNA with completely unexplainable origin. And this DNA should have a very important information and should possess very important information and should, of course, be not explainable how it's appeared, this DNA. The problem, of course, is that she cannot find this type of DNA. And there is no even slight possibility to find, because I can see, and in my experience as a molecular biologist, I have been browsing into the human genome, into the mouse genome, and all kinds of genomes everywhere. You can pretty much explain any source of DNA, every single enzyme, any single gene, where it comes from, how it's appeared, and what actually happened. So to prove her hypothesis was that it will take a long time to start and even to look and to find such a DNA that it's not explainable to appear. So, but let her try. So that was about 5-6 years ago. So I didn't hear anything from her since then. Another very interesting kind of maybe lecture was given from this very young guy from Discovery Institute. I think his name was X.
Yeah, Douglas X.
Probably Douglas X.
I've met him in Seattle. I was last year there.
So, and what was interesting and what he caught my attention, I was listening his lecture, and his lecture was, I think was 3 or 4 years ago. His lecture was something like Good, Bad, and Ugly, based on a Clint Eastwood movie, you know. It's something like that was so, and he presented. By the way, as far as I understood, he was a genuine student in Harvard, and he presented the data from Harvard University that he performed with the other students, probably PhD student was during that time. And his idea was that if you mutate just Okay, maybe it's easier to explain when I try to describe what was setup of his experiments. In that case, you can understand much easier what I mean. So bacteria have a resistance due to some enzyme, which is a cellular enzyme. It is on a wall, on the bacterial wall enzyme. And they try to mutate this particular enzyme just with very small point mutations. They try to mutate in a bigger portions. They try to mutate in a much higher, bigger, larger portions. And they try to figure it out how this bacteria, once it's mutated in different portions, will recover and will regain the resistance against the particular antibiotic. So am I clear about that? So that's probably very clear. And his sound was that this is a super unique experiment, something very kind of never been done so far. In fact, we are doing those kinds of experiments almost every day. I mean, this is the classical deletion mutant experiments where you try to figure it out which particular section of the gene or the protein is important for some specific function. So once you make a deletion mutants and try to see how the phenotype behaves from the different deletion mutants, you can figure it out actually which section of this particular protein is important, is involved in different processes. So it's as simple as that. But I was very surprised once he started to analyze this data, and suddenly he said, okay, look at that. So the nature is good to make another, to make a function, to make a new information when you delete a very small, like a point mutations. But this enzyme cannot recover and cannot make the bacteria cannot regain information, new information, cannot do that if you delete too much. So therefore, that was the ugly side. And his comment was that nature by itself cannot de novo make information. So that was his idea. And initially, I was kind of, before, when I was listening this talk, initially I was kind of, I knew that something is wrong here, but I didn't know what exactly is wrong. And just because I have a very good experience with all of those kinds of experiments and everything, I see where the problem was, in fact. So what I saw here was very legitimate experiment, very well performed experiment, and he even showed the published papers, 2 papers from this particular study. But when you start to analyze, you see that he provides completely wrong model for evolution. So what is the wrong model? The wrong model in his case is that the cell should make information out of nowhere, of nothing, and just put it, copy, and just paste it in the losing site. In that case, the information which is lost during the deletion should be regained back. And he, of course, didn't observe that. Therefore, there is no de novo information appear. That was his particular conclusion, which is completely wrong model. The evolution does not work in this way. So it's a very kind of interesting to see that sometimes the scientists, I'm sure I can call this scientists pseudoscientists, they're trying to provide a wrong model, but using this wrong model, they try to show that this wrong model didn't work. Meaning, of course, if it's wrong model will not work. Therefore, the people will have some sort of feeling that actually, indeed, he is correct. But in fact, he is not correct because the model is wrong. The nature does not make information like out of nothing. And in fact, the evolution does not work in this way. This is not the mechanism as it works. So the new information, usually it's appear and the mechanisms are very well known. It's called duplication, translocation, mutations. So when you have already some gene, this gene could be duplicated, could be put in another spot, and can start to this second gene could be subject of modifications and mutations, and of course can gain new information. But he didn't mention that. He just every time when he is making deletions of his mutant, he put the bacteria in exactly the same conditions and tried to figure it out. Is it possible in that case because to have new information? Of course, it's not possible because it's not possible. The things that are not possible never will happen. That's it. But having this particular example is very interesting example because having this example, you can see how actually the model is, how the model works. If you have deletion mutant and put in the same conditions in the same environment, the bacteria will die, of course. It will not survive. But the evolution works in different way. If you have deletion mutants, for example, there might be conditions, there might be around another environment where this particular deletion, for example, might happen to be advantageous. So might happen to be very suitable for surviving in the new conditions. That's how the evolution works. It's not like having generation out of nothing of information out of nothing. It's not working this way. So this is my second example where I can explain and to say that sometimes by providing a wrong, not correct model, you're trying to disprove this model. And of course, it's easy to disprove wrong model because it's a wrong model. There's no doubt about that. Back to my experience with the dispute with Gneiss, this the first time that I told you my first one of my first debates with creationists, she said, "Oh, you know, there cannot be macroevolution because in order to have a new species, you need to have at the same time several mutations to occur, and all of these several mutations to be in the right direction, to provide the right direction." That sounds very logical to me, but it's not true. It's a wrong model. Simply, whatever didn't, it's not possible to happen several mutations at the same time to occur in the same moment, and that to give you immediately a new species, for example. The speciation, the formation of new species, is a process of gaining of new features by mutations and adaptations and selection. It just takes more time. So my point was wrong models are very frequently presented from Discovery Institute in order to disprove it and to show that the actual evolution and there is ID, intelligent designer. Also, I want to say I'm not person in any kind of any way to say is there intelligent designer or there is not intelligent designer. That's not the goal here. What is the goal here is to show that everything that we observe as origin of life and the variety and everything could be explained naturally without need of specific intelligent designer. So this is a different that to say, yeah, there is no intelligence designer. We cannot say yes or no about that. I mean, it's not the subject of science. When I looked with your debate with Mike last time, 2 days ago, actually, 3 days ago, and he was talking about origin of genetic code, I think, during that time.
Yes.
And Mike was kind of very angry a little bit. He was trying to push too much. You tried to explain also what, I don't know his guy, and I don't want to know this guy, that he said that there is no chemical reaction between the amino acid and the codon.
And the anticodon, yes, on the tRNA, because they are at the two opposite ends of the molecule.
Yes. So I think you were correct in this way, that he, not you, he actually what meant that there is no direct interaction between the amino acid and the codon. Of course, there is no direct interaction. There is adaptive molecule, which is tRNA. And we know that. This is nothing that we do not know from the science. And he does not tell us anything new here. We know very well how the genetic code works, how the information is executed, and how the information from mRNA goes to protein. We know all of that processes, and to very, very high extension. So I don't see how his point is proving something about intelligent designer. There is nothing to prove by saying that there is no direct interaction between amino acid and the actual codon. So, but I'm open to listen you now. What is in your mind? Why you think that you can prove intelligent designer just by looking the genetic code and the formation of genetic code? What do you think?
First of all, I don't attempt to prove things. It's not about, science is not about proving things, but about providing the best explanation. And I agree with you, Dmiter, that science has a good knowledge. We know how the translation works, how the codons are assigned to amino acids. And that's precisely the point that I was making with Mike in regards of the distinction of operational science and origins science or historical science. So understanding and explaining how the cipher or the translation from messenger RNA and the genetic code to the amino acids assignment, how that is assigned, we know that. We know that 61 codons are assigned to 20 amino acids and 3 stop codons. So we know how that works. And that's exactly the operational science, which we have a pretty good understanding about it. Now, a completely different question is, how did that originate? How did it come to be? how did that assignment originate initially? This is a different question.
It's like archaeology. In archaeological study, you can go, you can see the artifacts, you can dig up the field, and you can see the artifact. You can guess what actually happened back in time, but you cannot say for sure, yeah, that was exactly how it happened.
Yes, exactly. That was my point. You are confirming what I said. We cannot prove what happened in the past. So whenever someone comes and asks me for proofs, then I will immediately say, we do not have proofs either that there was a creator involved or not. We cannot answer that question. What we can do is use abductive reasoning to the best explanation. When we see the evidence, then we can interrogate and try to find out what mechanisms explain best that feat. That's all we can do.
Okay, but there's always something but. Of course, we may never know exactly how, for example, genetic code appeared. We may never know exactly how origin of life occurred. But that doesn't mean we cannot provide natural explanation how could occur and how could appear. Meaning, if we are able to explain the entire process, if we are able to explain the entire process just using the nature, the regular laws, like chemical interactions, physical interactions, and show, even in the laboratory, show that the process in general works, it's feasible, it's possible, without some specific interference of unknown, this is already showing us that the process is completely feasible without miracle. It's possible. That doesn't say, oh yeah, exactly that happened, exactly like that happened. But it says that it's completely possible to happen in this way. Also, do not forget, science is full of surprises. We still might find very good conditions, and we can still regenerate the conditions of primordial or to some extent, and to show in laboratory conditions formation of primitive translation. It's still possible. By showing this entirely, showing the natural, in a natural process, you can directly prove the possibility of that naturally, without need of some sort of non-explainable event. So this is a totally different from to say, for example, oh, nobody can prove anything about the origin of life because it's happening by miracle. So I don't know what is your view. You want to say that in general we do not know right now how it happened, or in general we will never know how it happened? What exactly you want to say?
I think there are 2 points which, in my understanding, provide a better explanation leading to intelligent design. One is eliminative inductions, which means when science has no good explanation by natural means, or eventually even demonstrates that statistically it is very unlikely that natural means provide the feat in question, And the second one is when you have positive evidence, when you make a comparison of what intelligence can do, which non-intelligence can't do, and then look into the natural world and you see features and things which indicate a signature of intelligent design, of things that only intelligence can do. So you have...
Yeah, did you finish? I'm sorry.
Yeah, I think you want to. You have a thought or so. You can ask if you want.
So that's exactly why I try to explain that if you are able to provide, so if you are able to provide an explanation, experimental explanation and theoretical explanation, that origin of life, for example, origin of genetic code, origin of information could occur without specific, could occur entirely from what we know as a natural processes, as a chemistry, as a physics. You don't need anything else. Now, you said that have a signature of intelligence. So have a signature of intelligence, it's not meaning anything. It's just mean organization, mean level of organization. Signature of intelligence, it's not defined except level of organization, specific level of organization. So to say signature of intelligence, yes, there might be signature of intelligence, but that doesn't mean by itself that there is some forces which is not due, not explainable forces by chemistry, by physics, by regular interactions as we know. And I can, of course, prove it to you that you don't need a specific conditions. I mean, you don't need unknown conditions to have a specific synthesis or to produce something which is information, and to produce something which is actual living things. So what do you want to say about that?
Let me think. I mean, there are so many things to say about that.
So let me ask you something simple.
I think we need to elaborate what things that only intelligence can do, which random events cannot do, or statistically it is extremely unlikely that random events can do the same thing.
Okay, let me ask you something very clear. What is statistically unlikely to happen in origin of life?
Okay, the minimal genome to have a free living bacteria is about 1.3 million nucleotides.
Correct.
Okay, so if we posit that natural beings came with the right sequence to have that genome, the odds are 10 to the 700,000.
So
that is a huge number. You know, we have about 10 to the 80 atoms in the universe. But this is just a small problem. It goes deeper than that. If you have the nucleotides to get that genome, these nucleotides on the prebiotic Earth, rather than complexify and starting to polymerize, they would simply disintegrate. And Steve Banner has written a paper on that in 2012, The Paradoxes of Life, and he mentions the asphalt problem, which is that molecules, they disintegrate in a rather short time. And you know that RNA and DNA is a complex molecule made of 3 parts. So how do you solve that? Because there was no selection mechanism, and you need to select the right molecules which are used in life. And DNA and RNA was not simply laying around on the prebiotic Earth. We know how complex it is for cells to synthesize them. It's enormously complex, and special to make DNA. The ribonucleotide reductase enzyme is enormously complex, and it is essential to transform RNA into DNA. And DNA, we know that it is extant in all life forms. It had to be there when life started. So how do you explain that? It's enormous complexity.
Could you hang on a minute, Otangelo?
Also, I just want to say that from my previous experience, Mike at that point was very angry and he tried to silence you. I will go just to the bathroom for 5 seconds and I'll be back because too much coffee this morning was. So
okay.
Just give me, yeah.
Otangelo, you just mentioned a paper. Could you please put the name of the paper and the year and the author in the chat so that we can look it up?
Yes, I did put the link. I did put several papers. The first was about gene duplication. It is up there with the link. Then about the origin of the genetic code, I put the link as well.
Oh, you did? In private chat or in
both,
the comments?
Both, in the private chat and in the comments chat as well. Do you want that I repost it?
Okay, I can't see it in the regular chat. Let me see.
I'll repost it. Just a moment, okay?
I see one of them in private. Oh, I see 2 of them in private chat. I can. You have a wrench, so I'm sorry. I didn't realize that you'd put them in chat. But I need to.
Both, but I will repost it.
Okay. Because I want to put those in the description.
Is this a good time to kind of jump in here?
Sure. Have you guys met before, Dr. Alchemy and Otangelo?
I've just talked to him in the chats, never directly like now.
Okay.
Yeah, we've had
the origin of and evolution of the genetic code, the universal enigma from Eugene Koonin, and that's the link. It's a very well-known paper.
Okay, let me see here.
Thank you for that. So Otangelo, I was watching. I haven't finished it, but I was watching your video from about a week ago, where you were tackling kind of the logic and the reasoning and argumentation that you're using and that your opponents might be using. Can you summarize your argument, not necessarily the actual syllogism, because I know you and I have an ongoing conversation in that respect. But is your argument deductive, inductive, or abductive? Or is
abductive. It's abductive.
So you acknowledge that it's an abductive argument.
I need a minute. I need to open the door to my wife. Just a minute, please, okay?
Sure.
I will try to pull out my papers and several papers in that case.
While Otangelo has stepped away, kind of for your guys' purview and review, what I see as the simplest argument for abiogenesis by naturalistic means goes something like, at one point in time, historically, the universe was sterile, meaning no life existed in any form. Life exists today on the planet Earth. Conclusion, at some point, life arose naturally. So, this is kind of the simplest inference for naturalistic abiogenesis. But if you acknowledge that historically there was a time where life didn't exist anywhere in the universe, and if you acknowledge that life exists today, you're left with naturalistic mechanisms. Most theists, if they take the time and think about it, they won't accept the first premise, because from their perspective, God was in the universe even though it was sterile. So as a biologist, that doesn't make a whole lot of sense to me. Life is a term we use to describe biology, chemistry and physiology. What do you guys think of kind of that simple inference?
Yeah, I will tell you exactly what the life is. It sounds too self-confident, but I think I can say it. I know what you mean. So, but let's start. So I will give you. So here, let me see my. Just give me 1 second. Share a screen. So if I want to share screen, I can share. Let's see, can I do that? Okay, obviously I can.
Are you having trouble doing that? Let me see. Oh, good. You're good.
Can I share it? So
you should be sharing. There you go.
oh
You're
yeah,
on.
yeah. Okay, just 1 second. So yeah. Oh, good. That's nice. Yeah. Okay, so here is the deal. So, can you see that?
Yes.
Otangelo?
Yes, I can see it.
All right. So this is what I'm showing you is, first of all, the things that you are going to listen now, today. All of the information that you will receive today, especially my first 2 papers that I published. Those papers has been, I'm watching how many people downloaded these papers. And at the moment, they're about, let me say, 1,000 people in the entire planet Earth. So in compare with several, probably hundreds of thousands that was in the subject, in the field. And mostly when you're talking for origin of life, most of the papers are somehow linked or dedicated to the Rna World Hypothesis. So hundreds of thousands of viewers, people, scientists that know about Rna World Hypothesis versus whatever I'm going to show you here, it's relatively new. So my first paper was published in 2018, and the second paper is published 2020, this year. And this is a view from my third paper.
It's way too happy.
Yeah. So
you want to mute Otangelo?
I can't do that because I'm not the.
I don't know how to mute. Oh, unconnected.
Did you just say you don't know how to mute in your own stream?
No, yeah, I asked him if he wanted to mute. I don't know how to mute, apparently.
Wow.
Well, how did you mute? Oh, mute mic.
Yeah, that might be the one.
It's that mute mic button?
I'm assuming so.
I don't know. That's way too simple for me.
Yeah.
So Dmiter, do you think I was angry with him the other day? I was trying to be forceful and keep him on task.
Yeah, I know what was. It's fine. It's everything. Yeah. Okay, you were reasonable. I'm not saying that you're not reasonable, of course.
Well, I was being a little unreasonable. I felt like I was being a little too forceful.
I can say I have a relatively good experience with debating in any way. So you always have to look from the other side. I understand very well that. Yeah. So, I mean, sometimes the things looks very accurately from their point of view.
Yeah.
And we have to acknowledge that.
Well, my point was that John Maddox, when he said that, he was trying to pull one over on somebody who didn't know a lot about molecular biology. So he didn't know a lot. So he was trying to pull this one-upmanship. And he was trying to say that it was not deterministic. And it not being deterministic is quite a stretch. I mean, that's a little much.
Yeah.
Oh, molecular alchemy. What happened to you? I see you. Oh, you're there. You just
I was going to ask you, for those of us who haven't seen everything related to what you were just talking about, what exactly did Maddox say?
Oh, he said he made the comment that it was not that deterministic chemistry had been disproven in protein translation. That was big. And he got that comment from a David Abel paper, and David Abel is highly suspect. So anyway, so I was just really put off by that, and sort of put off by the fact that he was taking advantage of Godless Engineer and his knowledge. Oh, I'm sorry, Otangelo, you're back.
Yeah.
Okay, I'm going to shut up.
So before starting to explain this particular figure, let me see. Oh, can you see my pointer there, or you cannot see? So
I can see it too. I can see it.
You can see it, okay. So before starting to explain this figure, I will say just thinking for what you just said for the complexity. First of all, by what model? So by what model, for example, you said that the actual probability to have super huge genome, super big the protein, by what model supposed to appear as a super big protein?
Well, I don't know what you mean by model, but my proposition is that cells are irreducibly complex and they cannot evolve chemically by slow processes during very long periods of time, because you need three basic elements that work together, which is matter, energy, and information. And if one of these three is not there, you cannot create and kickstart self-replicating cells.
Let me ask you something. I'm sorry, did I disrupt you? I'm sorry.
No, you can go ahead.
So, can I ask you, do you know what PCR means?
I think PCR is artificial polymerization, no?
Yeah, it is polymerization chain reaction.
Mm-hmm. So, but this is using by intelligence.
So, if you perform PCR, you're using polymerase. The polymerase is an actual Taq polymerase. It's isolated from Thermophilus aquaticus, I think was, organism that is living in about 100 degrees Celsius. It's a very hot conditions. So isolation of this particular enzyme, you can create the right conditions and you can do a polymerization reaction. And this polymerization reaction is a widespread method tool for everyone in the molecular biology and a diagnostic as well. So this particular example show you that you can take elements from another cell, you can take proteins, activity, and put this activity and proteins in a tube in laboratory conditions and to provide and to show that it works independently of anything else. Also, that's true not just for this polymerization, it's true for any single protein that I have worked with. You can take this protein and put it in separate conditions and show how it works independently of anything else around, independently of the conditions of the cell. Meaning, you can reconstitute any process, transcription, translation, replication, everything, you can make it separately in a tube and show how the process works. And that's what we're using in experimental conditions. So to say that it's irreducible complex, that's not true. It is completely well reducible. You can reduce all the functions in the cell and make those functions to work independently in the tube. So it is not irreducible, it's completely well reducible. That is the fact from the practice, experimental practice. That's how we are working with. Now, when I'm trying to say what actually by what model is, I mean, if your model is, for example, let me say you have prebiotic conditions. And in this prebiotic condition suddenly appears a cell, fully developed cell. If this is the model, of course it's not possible because, as you say, if you calculate the probability those things to happen, the number is very big, probably bigger than the elementary particles in the entire universe, meaning not possible. Even simple protein, bigger than 50 amino acids, if you're trying to arrange statistically by chance, it's impossible because the mathematics is very clear. And you're right, the mathematics is clear and shows that it's not possible to have protein fully developed suddenly appeared. There's something else. Even if you assume that you have fully developed protein in a prebiotic conditions, that means nothing because this particular protein, if it's appeared out of nothing, this protein should do something with the other proteins, with the other molecules around, which are not there yet. So floating by itself, very well developed protein in the prebiotic conditions will do nothing. It simply will disappear. Which means that the model, it's not the model that the scientists think about. So I'm working in the molecular biology probably around maybe 20 plus years. In the origin of life, I am already maybe around 6 years. Maybe I already read probably about 500 papers about origin of life, professional papers. And I never seen even single element or even single sentence that says that the origin of life should start with very well developed self. It's simply not the model that we scientists work with. We are working with something very small, very simple, which start to become more complex and more complex and more complex. And then, due to the Darwinian evolution and selection, processes can become more and more complex. Simply, simple and statistically probable can grow, and with time and selection can grow in something which became bigger and bigger. Now you will say how it's possible to have, for example, nucleotides. The answer is very simple. Again, it's called selection. If you have selection towards something, you will select this particular nucleotide. You can say how it's possible information to appear out of non-information without having intelligent designer, for example. And that's a legitimate question. Personally, I asked myself many times. However, you can find a very clear process of selections which use non-information sequence, which doesn't have any information, and this sequence to be rearranged and to happen and to be selected as information. Now,
okay, I would like to answer to that, Dmiter. First of all, there was no selection in prebiotic chemistry, and I have made a video in regards of the supposed chemical evolution of amino acids and proteins. The video has about 20 minutes. If Mike would like, then he could eventually put it for play, and then I will expose precisely what I think in regards of that subject. Mike, would that be possible? It's about 20 minutes.
Just 1 second before starting. You want to show how it's not possible to have a selection in prebiotic condition? Is that what you want to say?
Yeah, I would list all the problems which I see in regards of chemical evolution of amino acids.
Okay. So, Mike,
can you put it? It's in here in the chat box. The link, Mike.
The chat. I put it in the description.
Link it later. Link it later. Like disrupting this for a 20-minute clip that will work.
That's going to be a little difficult.
Can you put a link?
Yeah, I put it. I put the link.
Otangelo, highlight the top 3 reasons that you can give that supports your statement that, quote, there was no selection in prebiotic chemistry. I typed it out as you were saying it. That's a strong statement. What are the top 2 or 3 reasons you have that proves or demonstrates that selection didn't occur there? First of all, define selection in this context. What do you mean by that phrase?
Okay, there are some papers which were published recently which claim, I don't know if it is true, that there were supposedly about 500 different amino acids extant on the prebiotic Earth. We know that life uses 20, in some cases 22 amino acids. So you need to join them to the building site of the cell and select them from that pool of different amino acids supposedly existing on the prebiotic Earth. I see no reason why molecules would have the intent to select the 20 amino acids used in life. That would be an example. We can extend the same problem to nucleotides. As you know, nucleotides, they are made of the base, of the backbone, of the ribose, and the phosphate, and they are joined together at the right place at the backbone. So the base, we use 4 bases. There are used 4 bases. Why are there just used precisely pyrimidines and purines with the composition of carbon and nitrogen, which provides then the Watson-Crick base pairing and the right forces to have the base pairing, which must be just right to have the hydrogen base pairing. So that composition, which is actually something that I brought up with my debate with Leophilius, there is no reason why that composition has to be the way it is. The molecular, the atomic, the elemental composition of nitrogen and carbon, there could be sulfur or whatever element you want to compose the pyrimidines and purines. Why is it precisely that composition that there is no physical necessity in order to be so? So there is no reason why these components were selected to make these bases.
Okay, can I answer your questions?
Yeah, go ahead.
First, when you're saying those kinds of things about not possible, there is no reason for. It's kind of holding out of not firmly confirmed, not firmly you can stand behind those, and I'll explain why. See, in the field of science of origin of life, I would say you may have some reason to say that. Probably if you said it about 10 years ago. 10 years ago, there was no modern theories, hypothesis for origin of life. 10 years ago was dominated by Rna World Hypothesis. So Rna World Hypothesis, it says that the first RNA started as a very little sequence. I'll be very short here, very short. But Rna World postulate or hypothesis is that from very short RNA first appeared the ribozymes, which is ribosomal enzymes, and those ribozymes are developed to provide and to was selected to the stage of self-replicating ribozyme, and that self-replicating ribozyme further was selected to the state of primitive translation and eventually formation of translation system. I was very simple here, just without details. But this hypothesis is very well questionable right now. At the moment, there is a very new brand, I will say a new generation of hypothesis, which does not stand immediately for RNA world. They stand for interaction between short peptide and RNA. And I will explain you later my hypothesis, which is different from the others. But that's not the point. The point is, I've been reading many papers, which first of all, it shows the prebiotic synthesis of nucleotides. For example, the experiments from John Sutherland, for example, and Dmiter Suslov experiments shows nucleotides to be very well pre-synthesized in prebiotic conditions. There is another experiments as well showing that in one pot chemistry or on several pot chemistry, you can have nucleotides as a prebiotic synthesis. Now, how it's possible to have particularly this nucleotide to be selected for the purpose of carry on the information? This is also very well explainable. And you have a very good reason for that, because you have a hydrogen bonds between purine and pyrimidine bases. And these hydrogen bonds maintain the structure very well. That doesn't mean there is no hydrogen bond in the other moleculars, but that means that those was happen to be suitable for formation, for carry on the first type of information. And ever since, I have never seen any paper that said that you cannot select, there is no way to select, there is no reason to select nucleotides. I've seen many papers which shows, yes, there is a huge complexity in prebiotic world, a huge complexity. For example, you do not expect to have just RNA. You expect to have forms of RNA, different modifications of RNA. You can expect to have RNA-like polymers. All of that is quite possible to exist. But the reason is why this particular RNA are selected for is because of the function that they carry on. And this is a function versus natural conditions, meaning the possibility to resist the particular environment and to carry on the mechanisms and reactions in particular environment. Now, also, if you say, do we know exactly what was the prebiotic earth conditions? Prebiotic earth conditions is just postulated. We can simplify prebiotic earth conditions. We can guess based, it's an educational guess based on everything that we have. How was the prebiotic conditions? We do not know exactly how was the prebiotic conditions. Therefore, you cannot say something like, oh, you cannot select that for. There is no reason to say this. The statement is incomplete. I think whatever you read is just the first part of the sentence, the first part of the paper. And in fact, I have a paper here that shows very clearly why, for example, the 20th of amino acids are selected for life. I have this paper. So there is a reason for that. There is a reason for this.
What reason is that?
The reason is the actual function. So you have part of the amino acids are positively charged, part of the amino acids are negatively charged. They cover the complexity of the functions everywhere. And through the selection of the process, you have particular variety of functions that can be generated based on this particular amino acids. For example, amino acid that carry on sulfhydryl group, SH group, can form a sulfhydryl bonds. And that's a function specifically for this particular amino acid. Another is positively charged amino acids like arginine, which is very good in interaction with RNA, which is negatively charged. Now, you see there is a specific function in each of the amino acids which is selected for. And there is no any way, there is no any reason to say that you cannot select this particular. No, it was selected exactly because of this particular group of this particular amino acid.
The selection in itself, it also carries the meaning of intent. When you select something, you do select it for a purpose, but molecules on prebiotic earth, they did not have the intent to have a specific function. No. They were just laying around.
I see what you mean.
Can I chime in here for a quick second?
Sure.
I think the word function in science, this would be not just biology, but more areas of science, physics, chemistry. I don't think that the term function or selection, or whichever one it was you said, I can't remember, it was one of those two, has the same meaning, that sort of intelligent agent connotation attached to it, like we use when we're colloquially speaking about selecting for something. It's sort of, I'm choosing this. I don't think that it's a choice being made section. It's sort of a process that ends up favoring something over another due to particular conditions that may be present. So it's sort of a selection, but it's not like there's some agent there making a choice. So I think that just comes down to how the word selection is being used within that particular context, within the context of molecular biology and origins of life.
Okay, that was nice. However, there is only one purpose and intention of the moleculars as a function. It's called surviving.
Well, but molecules, they are not alive.
These
are just chemicals. Molecules, they are not alive. They're just chemicals laying around on the prebiotic earth without the urge to complexify, to self-replicate, to become live, to become cells.
I understand. I understand exactly what you mean.
And the asphalt problem, which I mentioned earlier, is a very, very big problem in abiogenesis research, because molecules, they do not complexify, they degenerate and become asphalts. And this is a point which Steve Banner very clearly stretched out in his science paper, The Paradoxes of Life.
Okay. Can I continue or just...
Yeah.
Did you finish?
Yeah, go ahead.
So, I will show you exactly right now, demonstrate you how the selection in general works is called stability. For example, I'll give you example. This is just example. What is base pairs in nucleotides? So you have Watson-Crick base pairs. GC base pairs are held by how many hydrogen bonds?
2 or 3. I'm not sure now.
Okay, 3.
Okay.
So you have 3 hydrogen bonds which maintain GC pair. Correct? Everybody agree with me? If you have RNA, in RNA you have A and U, adenine and uracil. So how many hydrogen bonds you maintain this particular base pair? There are only 2. So the GC-rich RNA, if you have base pairs, let me say it's double-strand RNA, short, let me say 5 base pairs and 5 from the other side, 10. So 5 from one side and 5 from the other side. One is GC and another is AU base pairs. So you have sequence difference. One is predominantly GC, another is predominantly AU. Now, if you hit these 2 different types, different sequences of RNA, you just hit it in, of course, in solution. What will happen?
You say it.
Hmm?
You say it. I don't know.
Okay, so I'm trying to explain here. So it's the process of melting. So the hydrogen bonds will disrupt and the RNA duplex will melt. So they will become single strand. The temperature of melting, it's called TM. TM, temperature of melting, will be quite different between AU-rich RNA and GC-rich RNA. Why? Because of the number of hydrogen bonds. GC-rich RNA, it's simply more stable than AU-rich RNA. And see, right now I don't need to prove it. That. Why? Because this is experience and experimental research, maybe from 70s. We know that. It's proven millions of times in many experiments. Every single PCR actually do it. Now, the temperature of melting is different, meaning the point where you will have completely melted GC-rich RNA will be slightly higher than completely melted AU-rich RNA. This is physics, pure physics. Do you see any intelligent designer in this particular physics?
No.
No, I also don't see. Now, do you see a selection process here?
You could say so, yes.
I did.
I see very clear selection process, and that was just example. You have selection of GC-rich RNA versus AU-rich RNA. If you repeat that many times, it's quite possible, because you have also process of degradation of this polymer. So it's quite possible, because the GC-rich RNA, it's slightly more stable, as I explained just before a few seconds, than AU-rich, first will disappear or will be degraded AU-rich RNA than GC-rich RNA. And this is example for absolutely natural selection, which, by the way, select based on a sequence.
Now, the selection is based on the temperature to which the molecules are exposed.
Exactly, based on the temperature, but it's also based on a sequence of this RNA. One is GC-rich, another is AU-rich.
Well, the thing is that the temperature varies in a chaotic manner. It is not an organized temperature change. Climate change is not organized. So what we have actually is not a selection, but a reaction based on the exposure to the environment.
So did you see my point? That was a simple example. I tried to simplify this example in a way to try to demonstrate how a natural process can select toward a specific sequence.
Yeah, I disagree with the calling this selection. I would rather name it as a reaction based on the temperature change in the environment, and that reaction has not will not complexify the molecules into information-bearing molecule or in a function-bearing ribozyme or something that eventually later could be used to make molecular machines, and then even later a self-replicating cell. What you will have is a reaction of the molecule based on the environment to which it is exposed, but the natural trajectory of molecules is not complexification. It is just randomization and to become simpler and to become unusable asphalts.
Okay, I didn't explain you origin of life. I didn't mean that. What do I mean is to show you just that a specific natural process can select for sequence. It's an absolutely natural process. For example, as I told
as I said, Dmiter, I don't think that selection would be the appropriate word for what you described now. I would describe it as a reaction based on the temperature to which the molecule
That's what
is
selection
exposed.
is.
Well, that is the selection. So let me say you have n numbers of repeat up and down the temperature.
Okay,
Many.
the question.
So
okay, the question is, could that kind of selection lead to an increase of complex specification, which then leads to what we see in life?
Yeah, that's a wonderful question, actually. This is exactly what I asked myself 5 years ago. And I can show you how it happened. And I can show you how that could happen, entirely natural. So my point here was, in this particular example, is to see that you may have a situation where specific sequence could be more stable than the other sequence. That's what I'm trying to say here.
Well, I think we need to start at the atomic level. You have nucleobases, pyrimidines and purines in a very specific order of atoms, nitrogen and carbon, which needs to be in that order in order to have Watson-Crick base pairing and the right forces and the right distance of about 25 angstroms.
I know what
between, all this is.
Just 1 second. You can continue to the others. I need to answer the phone.
Okay.
Speed, say something. Let us know you're still alive.
I'm still alive.
I know you didn't croak over on me.
Barely. I've had several strokes, but I'm okay. I can still operate the mouse with my right hand with my index finger, but the rest of me is seizing up, but I'm okay. But anyway, see, the thing about prebiotic Earth is there were quadrillions, quintillions, sextillions of different microenvironments turning.
Oh yeah, the amount of different. What was happening on the bottom of the ocean was way different than what was happening on the surface, which was way different than what might have been happening in this ridge, because there were different chemicals there. And then, the ocean is churning and stuff gets mixed together. And the thing I think with people who just cannot see how the origins of life could have happened naturally, I don't know if they're capable of seeing or understanding just how chaotic and complex the early Earth would have been. And like you said, how drastically different environments could have been just within a square mile of, say, a part of the ocean. You could have had numerous different environments with numerous different types of chemicals, and boom, you start a lot more complex happening.
Yeah, and it's this is my pushback against reductive thinking. When you draw a diagram and you start with this reductive scientific thinking, you can be overly greedy in your reduction and not understand the reality. It's like when we take a cell and we start drawing these biochemical pathways, and we think that is what's going on in the cell. That is not what's going on in the cell. If you were to actually shrink yourself down and get into a cell, it would be utter chaos in there. And I mean, this reductive view of science becomes a problem. It becomes a problem in how we think about how things happen. Anyway, I'll shut up.
That's another thing is they have all those, the pictures. Okay. The pictures that they have and the animations of DNA replication and translation, transcription, all that stuff. Those are really, really cool, but they're like Feynman diagrams in physics. They're pretty cool, intuitive depictions of what's going on. But that actually looks like if you could somehow shrink yourself down and look at it. It's not going to look like that at all. It's going to be a very, very wild, chaotic, fast. You're probably not going to be able to keep up with what you're looking at, because that's how things work at the molecular and the atomic level. You're dealing with very, 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 very low mass things. They move quickly.
Now,
the forces that are acting on the strong interactions, electromagnetism, are all very strong at this level. Things do not happen slowly like, stuff that we see at the cosmic level. The stuff happens very, very quickly. It's very chaotic, but we can develop nice, cool little animations and pictorials to kind of intuitively describe what's going on after we've kind of sifted through all of the chaos. And that's exactly what those animations are.
The same thing happened to me with neuroscience. I studied neuroscience for about 4 years, and then I got an actual cow brain to dissect, and I started looking at it under the microscope, and then I saw an actual depiction of what a real cortex looks like, and it looked like this mass of head cheese, and it was like so complicated compared to the diagrams I had been used to, and I realized that I had the wrong impression from my textbooks of what was actually happening in the brain. And it's that sort of thing. You have to get rid of this reductive scientific thinking at some point and consider what is actually happening.
When we break reality down to molecules and atoms, throw our intuition out the window. And this is something that I think a lot of people on both sides, theist, atheist, agnostic, doesn't matter, don't want to do, because our intuition is comfortable. We know it. It's not something that's really novel. Having to throw that out the window when we go to understand something, that's why it's so difficult to understand, because it's non-intuitive. And reality at this level is quite frankly not explaining it, but we shouldn't take those literally. And I think that, like I said, a lot of people just either want to or can't throw their intuition out the window when we start to break reality down to its most fundamental aspects.
Yeah, it's called quantum mechanics. And it is, to some extent, could be explained only by mathematics, nothing else. Only mathematics can explain it. So, Otangelo, can I go back to your example?
Go ahead.
So, just following immediately what you said. In prebiotic Earth conditions, what kind of moleculars you may have, according to your understanding?
Sorry, I didn't understand your question.
What could be synthesized in prebiotic conditions?
In my understanding, by unguided means, absolutely nothing. There would just be chaotic molecules on the prebiotic Earth laying around, either in the ocean or on the surface of the Earth or in ponds, whatever. There would be just chaotic molecules without any direction into complexification in direction to become alive.
Yeah, but my point is, what kind of species you can have there? What can you have amino acids in prebiotic conditions?
I mean, we know with the Miller-Urey experiments, even in the more recent ones, by Bada and so forth, that 8 of the 20 amino acids, like tryptophan, which is extremely complex to be synthesized in the cell, have never been synthesized in these experiments. So
I understand, but here I want, particularly, just humor me here. I would like from you more simple answers, not just complicated answers. I
think that random events are completely impotent to come up with the basic molecules required to start life.
Okay, I will prove you wrong right now, but let me just go step by step. I don't want to rush because you will miss the point. So, do you think you will have amino acids of any kind in prebiotic conditions or not?
Any kinds? There are any kinds. As I said before, they claim, I don't know if it's true, that there were about 500 different amino acids extant on the prebiotic Earth. So, yes.
Okay, so I read many papers about prebiotic conditions, and I can tell you right now, Miller and Urey experiments has been done in a variety of modifications, a variety of analyses, and there is around 100 published papers of Miller-Urey experiments. The Miller-Urey experiment was just the first one in 1953, actually. Since then, has been done hundreds of experiments in the same fashion with different conditions, trying to figure it out what is possible in prebiotic conditions. And I'm talking about not just endogenous to the Earth, but also exogenous from Earth. So what is available in the asteroids, what kind of moleculars you can expect,
I have listed a link. I have put a link here in the chat, and I am listing 27 different problems in regards of nucleotide synthesis. I think 12 or 15 different problems in regards of amino acid synthesis, problem in regards of getting hydrocarbons on the early Earth, and also phospholipids. After our chat, you can have a look, and then subsequently, you can eventually give me an answer, a written answer, or we can have another chat, and then we can eventually elucidate one or the other point of this list. But
so
I don't claim that it is not possible that amino acids were extant on the early Earth. But the problem is to select the ones that are used in life, then there are 8 of the 20 which are used in life, which have never been synthesized in these experiments. And there is, I can just mention the more complex one. The tryptophan is very complex to be made by the cell. And then you have also the problem to the trajectory to go from a prebiotic synthesis to when a cell metabolizes the amino acids, which you know requires all these complex metabolic pathways. And that trajectory is an enormous gap to which science has absolutely no explanation.
Okay, I understand your point of view, but all of that, what you just said, is about next. That's why I ask you, let's go step by step. And so how complex is everything?
If we are going step by step and starting right at the bottom of the situation, then my question is, how did you get ammonia, nitrogen, and carbon already fixed and usable for life on the prebiotic Earth? Because as you know, the nitrogen has to be fixed, carbon has to be, there is the Wood-Ljungdahl pathway, the reverse TCA cycle to make hydrocarbons. These cycles were extant on the early Earth. So we have to ask first, how did actually these basic atoms be available prebiotically?
Because they were there when the Earth formed.
So I just explained to you that it is published very well, the synthesis, pre-synthesis of amino acids, of nucleotides, and lipids as well. It is,
But provided
it
you
is
can,
published.
I will leave my email, Dmiter, I will leave my email, then you can send me these papers. I am sure that I can learn something from them. Maybe there are things that I am not up to date yet, and then I will give my opinion on it.
Well, it is published. I mean, I understand you. Of course, if you pull out my papers, especially the second one, you will see, and there is let me say here. You can go down to the references and you will see, I mean, the entire paper describes all of that processes. But, for example, I'll show you here just a brief, very simple. So where was that? Oh, here, okay. So, do you see this? So this is a summary, a simplified. So I picked it up from the paper which is described here. It's very simplified. What you can expect based on a data, based on a provided data in a prebiotic earth for the distribution of the amino acids, for example. And they are not all amino acids here. They are just some, and I will say most of the amino acids, but not all of the amino acids. And to have a life, the initial life, you do not need all amino acids. Nobody said that you must have all amino acids in order to start life. You don't say that. You cannot claim that. So in the paper, in my paper, I showed the most probable amino acids that you can expect in prebiotic life. What else? Nucleotides. It was shown as well. Let me find the same, from the same paper. I will go and scroll down.
For some reason, you're not sharing anymore.
I could also provide a couple papers on.
No, I'm not sharing right now.
Okay.
I'm not sharing it right now. So I will. I'm trying to find, but there is a, from the same paper. You can go down and see. There is a experimental proof that you can expect to have nucleotides in prebiotic conditions. So go and look it.
So Dmiter, let's suppose that you are right, and let's suppose I agree with you, and we have these nucleotides in prebiotic conditions. Okay. So let's even suppose that you have a genome fully with the right sequence to make the proteins, the proteome required to have a minimal cell. Let's suppose you have the right sequence. What good would that genome be for just laying around on the prebiotic earth?
Nothing.
Yeah, exactly.
By your model is nothing. Because,
nothing.
Because by your model,
it would simply disintegrate slowly and then randomize.
Let me explain. By your model, you're supposed to have fully developed genome somehow. And this is your model, but what I'm trying to say that your model is wrong. We have never worked with that model. We have different models.
So what would be your model?
I can explain to you. So do you want me to explain?
Yeah, go ahead.
Okay, so first, let's go back just for a second. I will try to be relatively fast because it's 2 hours now. And I think everybody probably is tired. But let me show you first here this particular figure. And do you see the figure? You don't see, probably. I don't know. Ah,
So sorry, I was sleeping at the switch.
Okay, so this is a figure which is very simplified to show how Darwinian evolution works by itself. And this is from my 3rd paper that I'm going to publish. But this is nothing that the scientist does not know. This is just, it's a simplified kind of presentation. So if you have a source of information which determine particular another moleculars which have a structural function, and this particular structural function can simply harbor or can protect, let's say this way, or stabilize the informational moleculars in that case. See, this is a system. You can think about the modern variant. Modern variant is RNA and protein. So you have genetic code which determine the sequence of the protein.
Okay.
But this does not say RNA or doesn't say RNA or doesn't say protein. It just works in the information and structure. So if you have this system and put this system to environment, what will happen? 2 things might happen: destruction, completely degradation of this system because of environment is too harsh, or just because for some reason this structure and function could protect the information. Let me say interact, a protein or peptide can interact with the RNA, might protect from this particular environment. Am I clear here? Okay, so that might survive the system.
There was nothing to survive because these are just molecules laying around on a prebiotic earth.
Yeah, I'm talking for molecules. I'm not talking for anything else. So right now, you imagine that this information is changed somehow. Mutation happened. You know what the mutation is, right?
Yes.
Too many reasons. Because there is a rule. Here you have a rule which determines this particular structure. The particular structure will be changed as well. Am I correct?
Okay.
Due to the rule. You can think about, for example, mutation in mRNA, which will appear as a different amino acid in the protein, for example.
Well, Dmiter, the problem which I see I understand where you want to go.
Let me finish. Let me just finish. So, if you have this particular situation, the mutation, the next round, this particular system might survive as well or might die, might be destructed. So, this is the moment where you have a positive feedback from the structure to the information, where the information determines its own supporting structure. This particular system, it's called Darwinian evolution, because anytime when it survives, can be next time could be slightly more complicated than the first time. This is the only system that can secure slightly more complicated, slightly more advanced, slightly more adapted form next time. It's a very simple to understand that because you have only 3 components. information, you have appropriate structure and something that determined by a rule this particular structure. So this is how the Darwinian evolution actually works. Now, if you think about here, I'm not saying anything about how complicated is this system in order to work. Could be very simple and still will work. So now you can tell me what you mean.
The link between information and the manufacturing of proteins requires at least 25 extremely complex macromolecule complexes, like the RNA polymerase, like the ribosome, and all the other stuff which makes the error check and correction, and so forth. Of course, all that kind of complexity was not extant on the prebiotic Earth. How do you make a link between information, which is instructional, and the amino acids, which then should become the molecular machines? There was no link there.
Okay, I didn't finish. So, as I mentioned, did I say here how complicated must be the system? Is it said that must be from ribosome, fully formed ribosome, to fully formed protein? It didn't say. This is just a principle. It's just how, in general, Darwinian evolution works.
Yes, but there was no Darwinian evolution. There was no urge to protect DNA. There was no. I think we need to stop attempting to say that molecules had an intent or an urge to become complex or something like that.
I understand, but there's nothing to intend. You just have environment.
Exactly. Yes.
That's it.
Just environment and molecules laying around on the prebiotic Earth.
So if you have environment, this environment can destroy this particular system or can survive. Are you agree with me that if you have such a system, might survive or might be destroyed?
Yes, there are these two directions.
Exactly. So I didn't say how big it is, how complicated it is. I just say the principle, how in general this system should work in general. I'm not saying how complicated it is.
Yes,
now,
it
if I show you that just a few nucleotides, which is statistically possible to gain information, and just a few amino acids, 2, for example, amino acids are here. Do you think such a system could be generated from few nucleotides and few amino acids?
No, I cannot imagine it. I don't know how you would establish the link.
Okay, I'll show you. Just 1 second. Just 1 second to find the proper picture. Okay. And I need to go to the right. You were actually, you witnessed my presentation before.
Yes.
I just wonder why. Okay, so share the screen. I should explain you again this particular picture. Share the screen.
Oh, I'm waiting.
Come on,
Okay.
Speed.
It's time.
First word of your name.
I don't see the screen.
Are you sleeping?
No, it's oh, there it is. Finally, it showed up. I'm on the ball.
Okay. Now, see this particular picture I showed you the last time. So,
yes, I remember it.
Yeah. Okay. If you have a very short very short RNA, meaning short a few nucleotides. And this RNA, if you put, as I explained, if you start to heat this RNA, what will happen? It will melt, so to the single strand in a high temperature. And when you chill down the temperature, they will hybridize based on the base pairs. And that is a natural process of hybridization. It happened every time when you're doing PCR, for example. They are just in a lower temperature, they find each other and they hybridize. Also, it's very well shown experimentally back to the Yarus in 92 or 96, probably was 1996. It showed experimentally the process of aminoacylation. This is amino acids when it's aminoacylated in this particular for the hydroxyl group of the RNA. So either in a 5' end or in a 3' end of the RNA, you can have a process. It's a esterification. Process of esterification between the carboxyl group of the amino acids and hydroxyl group in the RNA. So, and this has happened in the modern organisms when you have charging of tRNA. The tRNA is charged exactly by aminoacylation, but the process of tRNA charging, it's guided by aminoacyl synthetases. This is enzymes which are doing that. Right now, what I'm trying to show here is that, in fact, the process of aminoacylation will be in natural, in the tube, could happen in the environment as well. The difference is it will not be guided at all, and it will be random. And that's what we can expect here. Yeah. So you have a random aminoacylated RNA in one site, and lots of RNA that are not aminoacylated. And all of this will depend from one thing. It will depend on the concentration of the amino acids. And that's it. Basically, every single amino acid have the equal possibility to interact with any single RNA molecule. Are you agree with me?
Well, this is a direct interaction, but we do not have a direct interaction in life.
I understand. I completely understand. This is just a step. I didn't finish yet. So, is there something that you do not understand from this particular figure?
No, go ahead.
So,
go ahead.
What would happen? Random aminoacylation. That's what will happen. You have heating process where you have melted here. Do you see the pointer? The point? So you no. Okay. So in a high temperature, see the high temperature, you have a single strand RNA with aminoacylated. So these rhombs and squares and triangles represents the amino acids. And the lines represents the actual RNA.
Okay.
So when it go chill down, they hybridize.
Okay.
This hybridization process leads physically to go in a close proximity. Some amino acids comes in a close proximity because their corresponding RNA hybridizing, and you have a process of, because of this hybridization, if you have two aminoacylated RNA, they might bring together the amino acids there. So you have a process of hybridization, and because they are aminoacylated, they might stay close together.
Okay.
Is there something that it's not understandable? I'm just trying to be very clear, the system. So, peptide bonds happens in close proximity in a situation where there is no water. So you can have a peptide bond. So in cycles of the Earth where you have wet cycle, dry cycles, which is very well explainable, you can have a peptide bonds because of the hybridization process of their own RNA, of the already aminoacylated RNA. Therefore, you can have a deep up peptide with 2 amino acids, 3 peptides, or if you have statistically much lower level of several, not too many, but several peptides, like 2, 3, 5 amino acid peptides.
Okay.
Could be synthesized in this particular way.
Okay.
Now, my question is, do you see some process here, something that it's not natural?
No, it's all natural. By what you are explaining, it's all natural.
Okay, good. Now, at that situation, you have RNA, you have synthesized it short, very short, I emphasize short peptides. And those short peptides are not just peptides, they're synthesized because of the hybridization process. They're facilitated because of the hybridization process. Correct?
Mm-hmm.
Okay, let's just scroll a little bit down. Do you see scrolling me down or not?
Yes, yes.
Okay, good. So, now, here is the statistic where it plays role. That you like too much statistic, right?
Yeah.
Now, when you have peptides, this is just represents 2 types of peptides. And as you see, those peptides are very short. They are no longer than 4 or 5 amino acids.
Mm-hmm.
By chance, and here emphasizing by chance, you can have peptide that incorporates positively charged amino acid.
Okay.
This positively charged, most likely, let me say, is the best example is arginine, for example. The arginine is very well known to interact with RNA. Why? Because it's positively charged. So you have positively charged arginine, which interacts with negatively charged RNA.
Mm-hmm.
Okay. Those kinds of peptides are just, we call it RNA binding peptides.
Okay.
Much, much rare, statistically rare, you might have a situation where you may have 2 different amino acids in this very short peptide. By saying short, again, 3, 4, maybe 5 amino acids, not long. You may have 2 amino acids, one to be positively charged and another to be negatively charged. For example, the B, if you see the example B here, you have a rhomb on the bottom, which is positively charged. On the top, you have triangle. So what happened? Because of existing rhomb, I mean positively charged amino acid, this peptide interacts with RNA based on a positive, negative charge. But also this peptide contains a negatively charged amino acid, which attracts positively charged amino acids. It just attracts based on a positive, negative interactions, charged.
Okay.
This creates a situation where slightly, I'm saying slightly more likely to have RNA which will be aminoacylated from this particular positively charged amino acid. See the proximity though. You see the proximity, and everything up to here is just positive in negative charge. Now, there is a statistic, I have to admit. There is a statistic here, and the statistic is down to 2 amino acids, and that's it. And just 4 or 5 amino acids. There's nothing else here. So statistically, if you calculate, it's a very reasonable statistic. Let me say 1 in a 10,000 interactions, 1 in a 10,000 peptides. One peptide might have these 2 particular amino acids. It's all statistic, and there is nothing else than positively, negatively charged interactions. So this second peptide, we call it bridge peptide. That's how we call it. Now let's go to the next figure, just to show you how magic happened. Okay, you see this figure?
Yeah.
Okay, this is the key. When I said everything so far, it's all about everything comes to this particular point of explanation. So, as I explained in the first figure, you have RNA that it's charged thanks to the bridge peptide, which is very simple. Bridge peptide, it's charged with positive amino acids, and this is in position 1 here. So you have RNA which is charged with positively charged amino acid. Now, and the same process of hybridization, peptide formation, as I explained in the figure 1, the first one, you will have many much more completely by chance, completely random aminoacylated RNA as well. And that goes in a process of hybridization dependent peptide formation, which is based on a chill down, heating, wet and dry cycles, which is very well understood. I mean, could be very well postulated in primordial Earth. Correct?
Okay.
I just want to be sure that you follow what I'm saying. That's why I'm so slow.
Yeah, I'm following you.
Okay. In the process of hybridization, formation peptides, which we call it HDP, those peptides are called HDP, will produce a numerous of all kinds of HDPs, different variants that doesn't have anything to do with what the selection is. They're just a random peptides. However, in one of those cases, sometimes, again, by chance, you will have reformation of bridge peptide, here in position 5.
Okay.
Now, this particular bridge peptide will enter back in a formation of new RNA, which will be charged in a new charged RNA with the positive amino acids. So it's a bridge peptide assisted charged RNA. It simply will enter in the same cycle again. So, do you understand that? Okay. So the aminoacylated RNA will enter, the new aminoacylated RNA will enter again in the same process. So you have a cycle of making de novo HDPs, de novo hybridization dependent peptides, which is very short, very random.
Okay.
Now, here's the point. This bridge peptide, because interacts with RNA, it stabilizes it, make it slightly more stable. This RNA with this particular sequence, it's slightly more stable, not too much, slightly more stable. Understand?
Mm-hmm.
I explained that before with GC-rich, with the GC-rich example.
Okay.
Also, it's true if you have peptide RNA interactions. So you just slow down the free energy by interacting with peptide and makes it more stable.
Okay.
In that case, the chance, the probability to appear slightly higher, to have this particular aminoacylated RNA, it's slightly higher next time because it makes it slightly more stable. The chance will increase very slightly. And enter again in the cycle of hybridization dependent peptide formation, as I explained by the cycles the first time. However, because the chance is slightly higher, you have in the system slightly more, a little bit more of aminoacylated RNA by bridge peptide. Therefore, you have a process of stabilization every time when this cycle goes from 1 to 3 to 4 to 5 and again 6, when you have again bridge peptide, goes back to 1. So do you understand this cycle? What I'm
Yes, yes. What I am curious to know, Dmiter, what will you have in the end of this process?
Exactly. That is a good question. You have slightly more every single cycle increase the chance of appearance of bridge peptide. Why? Because this particular aminoacylated bridge aminoacylated RNA will be presented slightly more frequently because the chance is increased every time. So you will have increase of chance to produce bridge peptide every time.
Okay.
Now, back to the picture that I showed you before. I showed you not because I showed you because of this moment, to understand better this moment. So where is the information here in this process? The information is in the sequence of the random sequence of this RNA. Now, you have to understand that amino acids like arginine, for example, usually interacts better with GC-rich RNA, and this is experimentally proved process. Therefore, a GC-rich RNA more frequently will be linked to arginine positively charged amino acid.
Okay.
Now, if you go back in the current situation where the modern biology is, so what happens I will switch off for just for a second. So what happened in the modern biology? You have tRNA, let's say tRNA here. Okay, tRNA. You have amino acyl synthetase, which is a protein. The amino acyl synthetase finds the correct tRNA and amino acylates the correct amino acid on the top. The structure of this amino acyl synthetases actually embedded the knowledge how to find this particular RNA, tRNA, with this particular anticodon, with this particular amino acid.
Yeah.
So the structure of the amino acyl synthetase determined by itself what kind of amino acids will be charged, this tRNA, with what kind of anticodon will be. That's why we said that the amino acyl synthetases are the only proteins which know the genetic code.
How do they know that?
Exactly. How do they know it? And I just explained to you one very simple system where you have a particular specific amino acylation of short RNA with specific amino. Just 1 second, I have a bird problem here. Just 1 second. Okay, I kicked him, so he wanna bite all of everything that it's rubber around me, and that's a huge kind of mess around. Hey, Mike, are you with me?
I am. I was just tending to a problem. What's up?
Okay, so now I explained to Otangelo a very simple system where you have a situation where amino acid like arginine, which is positively charged, could be specifically charged in a way that you have a specific GC-rich RNA. Now, your question will be immediately, why should be GC-rich? There is two major reasons. There is much more reasons, but I'll point two major reasons. First of all, arginine by itself as amino acid, it's already experimentally shown that interacts more frequently with GC-rich RNA. That's one thing. And second, as I explained, GC-rich RNA is simply slightly more stable in heat and those kinds of conditions, in hard conditions. And this is also experimentally shown. Now, you have information from this particular amino acid to the specific sequence of GC-rich sequence in this very short RNA. In the very short RNA. That creates a situation of Darwinian evolution, as I explained in the first figure, that you have information and structure. Because from that moment, as this system survive the harsh conditions, will continue to diversify and to become more complicated and more complicated. So if you pull out this particular paper, you will see what exactly I mean. In the second paper, it's explained the origin of the genetic code based on absolutely the same system. So you have a simple system, which is just 2 amino acids spaced by another 2 amino acids, for example, 4 amino acids. And very short RNA, which is just a few, let me say 5 to 10 bases. And that system could create a situation where the information began, because this is beginning of information purely natural. Information is the link between the amino acids, which is arginine, and this particular GC-rich sequence. So you have a situation of Darwinian evolution. This particular system is so simple that it's just possible. Now, of course, I can explain much more, but now we have 2.4 hours.
Yeah, we're pretty close. Can you, Dmiter, can you send me links to your 3 papers now? I don't think I have the 3rd one.
So the first one, nobody have it because I didn't submit it yet. So I submit it very soon. But you have the 2 papers.
Right.
And Otangelo also have the 2 papers. My 2 papers. You can go directly in Medline, and you can in the PubMed, and you type Koonin, and you'll find it.
Okay.
It's all simple.
Okay, I'll make sure I get links to them, because I have the papers, but I'm not sure where I put the links to the papers. So.
They're available. I specifically, when I published these papers, I was kind of very. I wanted this paper to be available, to be open, so everybody to be able to read it.
Yeah.
So Otangelo, just before we to close, because it's 2.4 hours now. And we can meet, of course, anytime after this week.
Sure.
No problem with more questions. I don't mind. But I want to say something just to have in mind. These 2 papers has been seen probably from about 1,000 scientists so far. It's relatively new. It's called RNA Peptide World. It's not RNA World. One of the problems of origin of life is exactly Rna World Hypothesis, because Rna World Hypothesis does not explain a lot of things, have too many gaps to be explained. And because of this, it's a very good source to be attacked. It's very easy to attack this type of hypothesis. Now, from probably around 10 reviewers, peer reviewers of these 2 papers, nobody so far from professional readers of these papers tried to say that that is not possible. I just explained to you a very natural process based on base pair interactions and based on a positive negative charge. You have a process of generation of information, which is so simple, a few nucleotides. I'm not saying huge information. We're talking here just for a few nucleotides and few amino acids. So not just simple, it's possible. Right now with my friend in Bulgaria, we are trying to set up experiment to show how this HZP hybridization dependent peptides will be produced, just as a proof of principle. I don't know what the result will be, but we're trying even experimentally to prove our point. No, it's a relatively new hypothesis. It's not very well known among the others, but we're gaining there. We continue.
Okay. Dmiter, it's very interesting to know a little bit more about your world. For me, it's new, really. I'm not used to these ideas, and it's a privilege to know all these things from you. And thank you very much for your very interesting exposition and explanation of these concepts. I would have to dive a little bit more into it to grasp a little bit more where it would lead. You know,
I would like, Otangelo, next time when we're chatting each other, probably thanks to Mike, come and disprove my hypothesis. I want you to put a finger in a place in my hypothesis and to say, because of something, that will never happen.
Okay. what I would like to invite you, Dmiter, when you have time, show up at my channel. I would like to play a video which I made in regards of chemical evolution of protein, origin of proteins, amino acids, so forth. I would like just to play it to you and to know what you think.
Absolutely. Just send me the link. You have my email as well, so you can send.
No,
me.
when you have time, come to my channel. I can give you the link. I can give you my email. Mike has my email. Let's just arrange when you have time to watch it, and then I would also like to know your ideas in regards of these ideas.
Okay. Yeah, I've got you guys on a joint email, so you should have each other's email. So you should be good. So
great. Okay.
Cool. It's been a long one and very, very interesting. Absolutely love your hypothesis, Dmiter. It's exciting.
I hope so. It's very simple. That's the most important.
Yep. And the creationists keep asking us for an explanation, and you certainly are giving them one. So,
okay.
So I'm gonna end the live stream right now. Is that okay?
Yeah.
Yeah.
Okay. And in the broadcast. So we'll see everybody who's still watching. We are